system. And just remember, endo means outside. Okay, so the endomembrane system is made up of membranes that have physical contact. Okay, let's talk about that. So you want to have physical contact or continuity. Kind of means touching. The other thing they can do is transfer things inside of membranes called vesicles. So you can think of these as when we talk about them like that, they're sort of like the baggers at the grocery store. Okay, so you put everything into little bags called vesicles. The big difference is our bags kind of close into bubbles. They're not open. So kind of like the baggers, which was one of my early jobs at Kroger's. All right. So here we have this endomembrane system. And that red arrow there is kind of what you want to pay attention to. The nucleus is sort of in charge of saying, here's the plans for a protein. The proteins are made by ribosomes. And then once you make them, your body's usually like, let's do something with this. And that can either be in the cell or outside the cell. Most of the ones that are in this endomembrane system are for export. Okay, so those are things that are likely leaving the cell. Now they can be used internally, but most of the time we sort of differentiate those by things that are leaving the cell. But the big thing is they're all going to be inside of a container, a membrane container. So this is just like you keep breaking off membranes. Okay, you keep breaking off membranes. Kind of like when you squeeze your toothpaste out, that last little thing that hangs on, you have to break it off. Okay? So we'll talk about the different types of this as we get. I could probably go crazy on just the picture, but let's get into the next section where we break that down. All right. First part of the endomembrane system, endoplasmic reticulum. Does anybody know what reticular means? It means net. There's actually a constellation called the reticulum. And so anything that has like a fine layer of network in it can be called reticulum. There's a sarcoplasmic reticulum in the muscle cells. There's there's some sort of an anatomy reticular formation, but I don't remember exactly where it is. But anytime you see reticular, that means like something with a network of fine structures. Okay, so this is really long to say and really long to spell, so we a lot of times abbreviate that as ER. Okay, not emergency room, endoplasmic reticulum. So when we talk about the surface area of all the membranes in a cell, this makes up a lot of it. Okay. So it's like a folded up net or sheet. So this sort of connects to the nuclear membrane. So usually you'll see the nucleus. I'll draw the nucleus in purple right here. That's a nucleus. Then you'll see the endoplasmic reticulum sort of hugging the nucleus. Okay, and this is very important because we look at like why is it there? Okay, and so remember that RNA is coming straight out into sort of the factory and then into the packaging system. And this thing is, some parts of it, which we'll get into this, are peppered with ribosomes. They're all over this thing. All right, so here's a much better drawing than mine. You can kind of see, switching to yellow here, there's a really tight interface between the nucleus and the ER. And they call it rough ER because it looks, I don't know, bumpy. And those, if you can't see them, those bumps are ribosomes, again, that are all over it. There's a section called the smooth ER, doesn't have the ribosomes. By then, the proteins are already made, okay? All right, so let's look at what it looks like. So again, okay, you're going to see these folded sheets really tightly put together. But the important part here is, like, what are these things? Those are layers of membrane. When we talk about membrane, we're usually talking about this kind of stuff here. Lots and lots of these. A phospholipid bilayer. Now, not all of them are exactly like that, but the majority are. And remember, these are self-organizing. If you disturb this thing, it will close up on its own. Okay. So, membrane, like that's what I was just talking about. Membranes are very fluid, okay? It's almost like a waterbed, or it's sort of like if you disturb it, it'll reorganize itself. So you can push into it, and it'll like memory foam that flows. Okay, I like that. Yeah, and that's what we mean by fluid. Fluids are not just liquids, they're things that flow. Like air can be a fluid. All right. Everybody okay? I went a little fast on that. There's smooth ER, which I already talked about in the cartoon. And what's going on in smooth ER? There's no ribosomes here. So these are responsible for non-proteins. No ribosomes, non-proteins. So you've got lipids and you've got carbs. Now, these also detox your cells, okay? So these will compartmentalize things that you don't want to mess up or damage or harm or whatever, things that are going on inside the cell. So this would be like, imagine you were at home in the kitchen and you spilled something nasty on the floor. Instead of cleaning it up, you just put like a sheet over it. Once you put that sheet over it, though, you can get rid of it. The sheet would completely surround it. It wouldn't just be on top of it. Not a perfect analogy. All right. Rough ER. We already talked about this. Ribosomes make it look bumpy. What do you predict that packages up? Yeah, proteins, because ribosomes make proteins. What kind of proteins? Proteins that your cells are going to secrete. Everybody understands the word secrete, right? It means like things that are coming out of your cell. All right. Let's talk about your Golgi apparatus, or Dictyosomes. I prefer Golgi apparatus. That's what everybody uses. All right. So what's the Golgi? Okay. It is parallel of these things called cristinae, and that means crest. You can almost hear the word crest in there. So you'll see the Golgi even more sort of stacked up like stacks of pita bread or stacks of tortillas. Most cells have a pretty good collection of these. It can be anywhere from 3 to 20. I'll probably never ask you that, but I just want you to know, like, it varies. What kind of cell would you think would have a lot of these? A cell's job would be what? A cell that would be making a lot of products, okay? A cell that doesn't really make a lot of products doesn't need as much of this, okay? So, from an evolutionary point of view, we just feel like this is a extension or, a furthering of the endoplasmic reticulum. So these guys are closely related. It doesn't have those rough areas like the endoplasmic reticulum does, but you'll see sort of like in cells, it sort of has like an in and an out section. Underneath my picture, you can see it there. This is where, if you look really close right here, this is where you see the stacks of tortillas, and you can see the vesicles starting to come in or out of it, and it's going to be different products in those. So usually you'll see things going from the ER and then out. So it definitely has like a sending and receiving side, okay? And I wrote ER under my picture, okay? Just like real life, I kind of get in the way. All right. So functionally, we've kind of gotten into this, and our function, the structure just like automatically leads to the function, okay? So you might see things in the endoplasmic reticulum be modified here. So something, usually an enzyme, will work on this, either build it up or break it down. Distribution. Now, not really meaning spread out, but meaning loading for transport, okay? This is sort of like, if you think of the ER as manufacturing, this is more of the packaging department. We're going to pack it up. All right, is that too fast? Okay, moving on. So the Golgi makes its own little vesicles that sort of squeeze off from the main Golgi. Okay, so these will pop off, and it kind of looks like this. Here's step one. Step two is you'll see this membrane extension, and then it pops off, and both sides close up. So the things from the, like we just said, the things from the endoplasmic reticulum. All right, we'll stop here.